if that's okay with you.
Yep, sounds good.
So we appreciate you joining us for our March iteration of our monthly acoustic seminar series, Norman. We look forward to hearing your talk. I think this may be the first time we've had a speaker talking about amphibians in general and frogs specifically during this series, so I'm excited about it. For everybody else who's here, thanks for joining us today, and we'll probably have a few more pop in here in the next minute or two, but I want to introduce this month's speaker. This is Dr. Norman Lee. Dr. Lee's at St. Olaf College in it's Northfield, Minnesota, right, Norman? Yeah. And I met Norman many years ago when I think this is right. I haven't solicited a bio for you, Norman. I think I understand it pretty well. I may be wrong, and you can supplement this if need be. But Norman was an undergrad working in the lab that I was postdocing in at the University of Toronto, Scarborough, many years ago. And he ultimately ended up staying in the lab with Dr. Andrew Mason and working on supplementing his undergraduate research experience with working on his PhD there, focused on sensory systems and auditory function. And I think you were working on parasitic flies at that point, right, Norman?
Yep.
And then Norman, after finishing his PhD at the University of Toronto, went south and joined us here in the States at the University of Minnesota, working with Dr. Mark B. And switched his systems to frogs, and he'll tell you more about that today. And then after I guess you were with Mark B for 3 years. Does that sound right?
It's closer to 5. I think it was 4 to 5.
Wow. So you were working with him for a long time. So he's got a lot to say about frogs. So that was his shift from invertebrates to vertebrates. And then after his postdoc stint, took a faculty position at St. Olaf, where he's at now. So wanted to share one quick fun story with him. I'll throw this out there. I know Norman and I haven't talked about this in a long time, but when I was a postdoc and he was a grad student, one year he came to a field site to join me in Oklahoma and had to transit the Canadian-US customs line. And he was bringing up a laser Doppler vibrometer, which is a device, for those of you not familiar with that, looks like a cross between an old school camcorder and some sort of device for non-lethal control of crowds. And he got hung up at the border with customs. And I think in some imminent trouble when he gave me a call. And I think at the time I was actually sitting in a dentist chair, getting a filling or something like that. So I had to remember having a conversation with a very suspicious sounding customs agent from a dental chair to try to extricate Norman from the clutches of our U.S. law enforcement as he came into the States to help with the research project. So Norman, I know that was a long time ago, but I wanted to share that with everybody because in case you had forgotten about that traumatic experience. But appreciate you joining us today here at UNH and for our community, acoustic community, and look forward to hearing about your science. So.
Thank you, Dan. Thanks for the introduction and thanks for the invitation to speak to all of you. That one lesson that I learned from that travel experience is to have the proper paperwork to enter the country. That was a lot of fun. Okay. Do we see my slides?
Yep, looks good. Looks good. Take it on.
Okay. So today I'm gonna tell you a little about some recent discoveries that we've been having with regards to frogs and how they're able to use their lung to filter out some noise that they are encountering in the environment. So you may have recently seen this in the news, how female frogs tune out useless, noisy males. And this was a press media piece written by Catherine Wu from The Atlantic. Anything that starts with before fret parties and frog ponds and breeding grounds with noisiest bachelors and lusty pools is going to be an interesting story to tell and to learn about. So this is work that Catherine Wu wrote to cover our recent publication that just happened last Thursday, which is on the ability of frogs to use their lungs to cancel out noise in the environment. So to describe this environment, let's imagine that we are in a noisy social setting, just like at this cocktail party. Let me get my laser pointer out. Okay, so at this cocktail party, you're in this party. There's all sorts of people talking at the same time. There are other environmental noises that could be of the clinging and clanging of the glass, could be the utensils being put down on a table. But your goal here is to focus on the conversation or the speech from this one individual. And the cocktail party problem describes our general difficulties that we might experience in being able to focus on the speech from this person or this individual. And if it's difficult for us with normal hearing, it's actually even way more difficult with people that have hearing impairments. So people that rely on things like hearing aids or cochlear implants, these types of technology tend to have difficulties in terms of being able to process the speech from a particular individual in a noisy social setting. So, for example, when in a noisy environment like this, for a human uttered vowel in a noisy social setting, what we find here in this old data plot is the frequency spectrum of this unaltered spectrum of a vowel sound. When we're producing vowel sounds, we tend to see these peaks, or these are formants that are peaks that are occurring at a regular frequency interval that are part of these voiced vowels. And within the spectrum, we also see that there is a considerable amount of noise that is masking these peaks, these formants. So one way to get around this, which is a typical strategy that things like hearing aids and things like other speech recognition technology will try to accomplish, is to accomplish this enhancement of the contrast between the spectral peaks of human speech from the spectrum of background noise. And here are some ways that we could accomplish this. We could either amplify those formant peaks to make them appear larger in amplitude relative to background noise, or we could also reduce the level of background noise with digital signal processing in a way that decreases the valleys to sort of emphasize the peaks by lowering the valleys of noise that's in between these formants. Or we could do both of those things at the same time. So it turns out that there are other situations that also encounter this cocktail party problem. So this is a human example of the problem, but we also have different analogs. For example, swarms of robots, right? So when they are making their decisions, they are in this environment where there are multiple robots. They're processing the scene that's around them, and they are having these decentralized decision points where they need to process that sensory input to be able to reach a particular target. But it turns out that animals are famous for having their own version of this cocktail party problem. We see social insects. In this case here, cicadas produce pretty significant amounts of background noise, and for the receiver of the signals, it tends to be difficult for the receiver to be able to process those signals individually. This also occurs in fish and in frogs and colonial birds and songbirds and bats. So this problem is not unique to humans. Each of these different animal taxa will have their own version of the human's cocktail party problem. So what I'm showing here is a family tree for terrestrial vertebrates. It's taken from a recent paper. And from this, what I'm showing in this tree here are that terrestrial vertebrates have inherited their sense of hearing from a common fish ancestor. So here we have this common fish ancestor that lived probably around 400 million years ago or so. So many of the basic auditory mechanisms are shared among the fish and terrestrial vertebrates, including us, including humans, because we have descended from this common ancestor. If we fast forward to about 300 to 360 million years or so, we have this lineage that is leading to the modern amphibians, reptiles and birds, and also mammals that have diverged since then as well. So in this scenario, we can say that the auditory systems of tree frogs and barn owls and gerbils, they'll have evolved independently from each other for more than 300 million years or so. And the critical point that I'm trying to make here is that this history has allowed evolution to derive novel mechanisms to solve common problems in different lineages. So let me give you an example. For example, maybe for the first 50 to 100 million years ago or so, right around here, after the divergence of all these early precursors of terrestrial vertebrates, they shared this common problem of having to hear on land, right? So there's this problem of impedance mismatching that involves transmitting sound from air in terms of a sound pressure wave to fluid motion in the inner ear. So in the early stages of this evolution, none of these land animals would have had tympanic middle ears. And it turns out that, in terms of this evolutionary history, that tympanic middle ear would have evolved independently, as shown in these ellipses, 3 times independently. And what's important to note here is that in these 3 different lineages, we see differences in the anatomical pieces that are being used to transduce air pressure waves into fluid motion. So for frogs and birds and reptiles, they tend to have a single auditory middle ear bone structure. But for mammals, we have 3 middle ear bones. So we're seeing different ways of addressing that same common problem. So I guess to sort of round off this discussion on the diversity of solutions, we're going to see that these different lineages have been exposed to different selective pressures that have allowed them to come up with different solutions to addressing this cocktail party problem. And the key here is to look at these different solutions to sort of understand the general principles that might help us solve that cocktail party problem. I'm going to describe to you the frogs that I work on. Here we have an image of the American green tree frog, Hyla cinerea. And these tree frogs, during the breeding season, they will produce a single note call. It's roughly 150 milliseconds long. And if we look at the spectrum of the call, there tends to be this 2 spectral peaks to the call. We have a low peak and a high peak. And when you hear this call, it sounds like a cross between the quack of a duck with the barking of a dog. And here's what it sounds like. As soon as I get the laser pointer out of the way. So that's a single note call. But in a noisy breeding pond where you might have hundreds of individuals calling at the same time, it'll sound more like this. So to a female that's actually trying to find an individual male for the purposes of mating, they have to solve this problem, being able to discern the calls of an individual and to select for the best one within a breeding pond. A little more about how the frog's auditory system works, and it turns out to be a little more unique compared to our human auditory system. We have in the frogs, there are these 2 auditory end organs known as the Amphibian papilla and the basilar papilla, and these are located in the frog's inner ear. So the basilar papilla is this little sensory papilla here, and the Amphibian papilla is located here. And this is quite different from our auditory end organ. We only have one of these, which is called the cochlea. So there's already this difference. And each of these end organs are responsible for encoding acoustic information from the two different peaks of the call. So the basilar papilla will be responsible for transducing the mostly the higher frequency peak of the call, and the amphibian papilla is responsible for transducing sound from the lower frequency peak of the call. And here we see a coronal section through the inner ear and the middle ear of the frog. Frogs don't have an external ear. When sound is arriving externally, it impinges on this eardrum, which causes the eardrum or the tympanum to vibrate. And that energy is transmitted via the stapes, or the middle ear bone, where it then impinges onto the inner ear, causing the inner ear fluid motion to then stimulate both the basilar and the amphibian papillae, depending on the sound frequency. Frog ears are also different from our ears in terms of how they are internally connected and coupled. So if we look at the MRI scan of a frog's head, what you'll see is the eardrum here, and then right behind that eardrum is a permanently open eustachian tube that then leads into this large mouth cavity. All of these spaces are air-filled. So when sound impinges on the eardrum, that sound pressure will cause the eardrum to vibrate, which will then have its force propagate through the open eustachian tube, through that mouth cavity, to the next eustachian tube, and have it work on the internal surface of the opposite eardrum. So the eardrum motion in this case here will depend on the difference in the forces that are acting on the external side of the eardrum, as well as the internal side. And it's this difference in the forces which causes this particular system to be known as a pressure difference receiver. And here we have an image of the mouth cavity. What is really cool about this type of a system here is that it's also closely connected, or closely interconnected, with the lungs. So the only thing that's sort of intervening between the lungs and the mouth cavity here is this glottal slit that's located around this region. So in essence, the air-filled lungs is interconnected with the open mouth cavity airspace, which is also interconnected with the eustachian tubes that leads to the eardrums. And in the early 90s, or around that time, people have already known this association between the lungs having an effect on the response of the eardrum. So here's an experiment that shows this association. These experiments were done on the barking tree frog, Hyla gradiosa, and plotted on the y-axis here are measurements of displacement using a device that called a laser vibrometer, which measures the vibrations of a substrate, and this is plotting the displacement of that movement. And on the x-axis here, we have sound frequency. When the lungs are deflated, so if we squeeze the air out of the lungs, we have frequency responses that are plotted in these dark black lines. Each of these lines represents sound from a different sound angle. And what we see here is this general frequency response when the lung is deflated. In this experiment, what they did was they used a syringe, and they were able to fill the lungs through the body wall with the syringe. And when they inflated the lungs of the frog and then measured the response of the eardrum to sound frequencies that are varying, what they saw was this change in the frequency response of the eardrum depending on sound direction. And in these types of plots, if we take the larger the differences are between these lines, it represents a measure of directionality. So how directional is that eardrum? In this condition here, what we see here is that there is a great level of directionality, or the eardrum is quite directional when the lung is inflated, if we're only looking at one eardrum. So the key here is these measurements are only from one eardrum. And if we superimpose the spectrum of the barking tree frog's call on top of this data graph, what you'll see here is a call that has a low frequency peak as well as a high frequency peak. And where there is a change in directionality due to this lung input, it happens at a frequency range that is not emphasized in this frog's call. So the paradox here is, if lung inflation does improve directionality, if it does make the eardrum more directional, why is it happening at a frequency range that is not really part of this frog's call? What good does that do? And that is known as our pulmonary paradox that we're trying to resolve. So in the early 90s, when those studies were published, they were done on a few number of animals. It might have been like 2 or 3 animals, and that was the data that they reported and published. And what we really wanted to do was we wanted to follow up on the study. We wanted to use a rigorous approach with a large number of individuals to carefully examine this directionality hypothesis, right? To carefully consider whether or not that lung input actually functions to improve directionality of the eardrum in a way that's useful to the frog. And we predicted that if this was the case, if lung inflation does increase the directionality, then we should see improvements in the directionality at sound frequencies that are part of the conspecific calls, if it's meant for improving their ability to localize conspecific calls. We also examined this alternative hypothesis. We also wanted to know whether or not spectral contrast enhancement is at play. Are the lungs affecting how the eardrums respond in a way that improves the frog's ability to hear its own conspecific calls? And the way that this might work is the lung to ear transmission pathway, when the lungs are inflated, that should either increase the sensitivity of the tympanum to sound frequencies that are part of the conspecific calls, or it should decrease the sensitivity of the eardrum to heterospecific calls, or both of those things could be happening at the same time. So we set out to test these two contrasting hypotheses using the Doppler laser vibrometer, that same one that I got caught going through customs with. And here we have, in this sound controlled environment, we have this laser Doppler vibrometer situated here. It's mounted on an air table. This device is a device that's used to measure vibrations on a surface, non-contact, so it's really useful in terms of measuring vibrations without touching the substrate. And what you see here is a focused beam of laser that's situated on the eardrum, so we can measure how the eardrum is responding to sound. The sound that we use in these experiments is a frequency modulated sweep. So that lets us test a range of sound frequencies starting from 200 Hertz to about 7,500 Hertz in a pretty short time frame. And so that's the frog on the pedestal, and right around this frog are these speakers that we can rotate and position at various angles around that frog. And then we would record the frequency response of the eardrum as a function of sound direction and sound frequency. So then we would do this experiment under different lung states. And this is an image, a composite image that shows you what the lungs look like in terms of the extent of the body wall when the frog is naturally inflated. So here we see the extent of the body wall, and right underneath this part of the body wall are the lungs that are in the frog. On this other side of the image, it's an image of the same frog, but with the lungs deflated. So we can see that it doesn't extend as far out as in this other instance when we have inflated the lungs. So the first thing that we wanted to confirm, or that we wanted to measure here, was, are the lungs actually receiving a sound? Are the lungs acting like a microphone? And the way that we did this was we had a speaker on the right side of the animal, and we were recording the vibrations of the body wall on top of the lungs in response to the sound presentation. What we found was that here we're plotting amplitude of the response and frequency on the x-axis. When the lungs were naturally inflated, we were able to detect a peak frequency resonance at about 1500 hertz or so. And when we squeezed the animal just gently, we were able to expel the air out of the lungs. We made that same measurement, and we were able to show that this lung resonance peak is diminished, and it's pretty much non-existent at this point. Then what we can do is we can re-inflate the lungs. And this is a pretty cool technique. You have a little modified straw with a pipette tip, and the pipette tip is put very close to the glottis in the frog's mouth. And then you can blow some air back into the lungs to re-inflate the lungs. And we were trying to re-inflate the lungs in a way that would provide this natural inflated state. And when we did that, we were able to recover this lung peak. So this set of experiment shows that the lungs, yes, the lungs do vibrate in response to sound, and it does so when the lungs are filled with air. So we really wanted to get at this idea of directionality. And how we do this is we wanted to determine whether or not the lung input actually helps in the directional hearing of this animal. And since frogs are bilaterally symmetric, they have ears that are laterally located on both sides, and we're assuming that the auditory system is symmetrical at this point, we can then calculate what's known as the interaural vibration amplitude difference. And we're going to call these plots IVAD plots. What this is it's really a measure of binaural disparity that represents directionality. And the way that you calculate this is you take a sound source and you put it on the right of the animal, and you present that sound and measure the amplitude of that vibration. And then you move that speaker around the animal, and you continue to measure those vibrational amplitudes. And then you're taking that difference in values. Let's say we have a vibration amplitude measurement at 60 degrees for that right eardrum. And we're subtracting from that the amplitude of vibration when the speaker is at minus 60 degrees. So we're assuming that the auditory system is symmetrical along the midline in front of the frog. And when we calculate that difference in amplitude, that gives us this indication of when sound is on the right side of the animal, the right eardrum will respond with a large amount of vibrations. When the sound is on the left side of the animal, the left ear will vibrate more so than the right ear. When we did this across the 3 lung states, inflated, deflated, and reinflated, when you look at this visually, they all look kind of the same, right? So there doesn't seem to be any differences, but it's really hard to tell with this type of a plot. So what we did was we took the difference between those IVAD plots. We assessed the directionality by taking the difference between the inflated IVAD versus the deflated IVAD. And that gives us this difference in amplitude plot. The y-axis is still sound frequency, and the x-axis that we're looking at is still sound presentation direction. And the colors here represents the amplitude of this difference that we see due to the lung input. And this might be a strange way to look at the frequency spectrum of a call, but imagine this to be a rotated frequency spectrum such that the frequency axis is on the y here, and then the amplitude, the relative amplitude, is on the x. What I'm trying to show you here is that for the green tree frog calls, there's this low spectral peak here and a high spectral peak here. And for both of those spectral peaks, if we look at this entire plot here, there really isn't much difference, right? The difference in the IVAD plots is roughly 0 decibels, which means that regardless of lung inflation, that lung inflation is not really affecting this measure of directionality. In other words, yes, when you look at an individual eardrum, you might see the directionality patterns that you would see in that earlier study in the early 1990s. But if you actually think about how that auditory system functions and take the difference between those two ears, those differences actually wash out to form this pale green plot. So I think from that data set, we can rule out this first hypothesis, that the lung inflation and the lung input is not really for increasing the directionality of the auditory system. So we had this other hypothesis left to test. And I will explain this plot to you in a little more detail. So we're looking at the role of the lung input on the sensitivity, the frequency response of the eardrum. And to do this, we presented sound from various different angles around the animal. So positive angles represents to the right of the animal. Negative angles represents to the left of the animal. And we varied that sound frequency from 0.2 hertz all the way up to about 7,500 hertz or so. And what we see here is that the eardrum is pretty responsive to a range of sound frequencies represented in color between 1 kilohertz to somewhere around 5 kilohertz or so. And if we're measuring these responses from the right eardrum. So sound at the right will cause that right eardrum to vibrate more intensely at this middle frequency range. As soon as we move that sound over to the opposite side of the animal, there's a reduction in the amplitude response of that right eardrum. We did this across the different lung inflation treatments. So the inflated state, and then the deflated state. Again, this is quite hard to tell the effects of that lung input on the sensitivity of the eardrum. But if you take the difference between the inflated state and the deflated state, what you get is this difference plot. And this difference plot is really striking because what it's showing us here is that there is this region there's this frequency range where there is a reduction in the amplitude of the eardrum's response when the lungs are inflated. So this region is roughly in front of the animal, right? So the frontal hemisphere of the animal, and this frequency range is somewhere around 1.5 kilohertz to a little over 2 kilohertz or so. This frequency range is actually overlapping with the frequency of resonance of the lungs. So that's a key thing to keep in mind here. So how does this particular reduction relate to the spectral content of the conspecific calls? Well, here's our difference plot rotated in a different way. It's the same plot. And now what we're plotting on top of that difference plot here is the frequency spectrum of the conspecific calls. This is the low peak. Here's the high peak. This box around this region is the region of overlap with the resonance of the lungs that we determined in that earlier experiment. And what we see here is a reduction in the amplitude response of the eardrum in between these two spectral peaks. So what we're seeing here is that the lung input is not affecting the two call peaks that are emphasized in the frog's conspecific calls, but there is a reduction that is happening in between those two call peaks. So what good is this reduction in the call peaks? What could this be for? And we addressed this question. We were trying to figure out, okay, so if there is this reduction, could it be for some biologically relevant noise that these green tree frogs might be encountering when they're within their breeding ponds during the breeding season? We turned to this North American Amphibian monitoring program. And this program is really neat. It spans more than 20 years worth of data collected by trained citizen scientists. These are people that took a quiz to ensure that they're able to identify different frog species based on their calls. And they would sample the different transects within the 26 states that this program was running within. And this database has descriptive calls for about 59 frog species, North American frog species. In that data set that we looked at, we found that there were 21,000 instances of co-occurrences with the green tree frogs that we work on. And among those 21,000 co-occurrences, there were 42 species that were found to co-occur or co-call in the same places that you would find green tree frogs. So when we also did this social network analysis to try and determine, of the 42 species, which are the top 10 species that co-occur with our green tree frogs. So our green tree frog is in the center of this network here. And these are the 10 species of frogs that are found to co-occur with the green tree frogs. And if we carefully look at the frequency spectra from the calls of these different frog species, it turns out half of the 10, so 5 of the 10 species, actually produce calls with spectral peaks that are directly in line with that range of amplitude reduction, which is also in line with the resonance peak of the lungs. So right here, I have these asterisks that are beside those 5 species that produce calls. Let's say, for example, this one here, there's a spectral peak here that could potentially be reduced in terms of its frog's percept of that peak with the lung input. And same for this Gulf Coast toad, barking tree frog, Fowler's toad, and etc. We're going to look at a few examples, 2 examples of this in a little more detail. So here's the frequency spectra of calls produced by Hyla gradiosa. We did this bioacoustic analysis where we were able to quantify this mean spectrum from this particular frog species. And here we see this frequency peak right here within that range of amplitude reduction that is mediated by that lung input. Plotted in a different way, here's a polar plot. So the way that you interpret this is the angles of sound presentation are from straight in front of the animal to the right of the animal, around to the back, left, and back to the front. And what I'm plotting here on this particular, on the radius here, are the amplitude reductions in response of that eardrum. And in dark red is the median amplitude reduction. So we're getting roughly 5 dB reduction of this particular peak in the Hyla gradiosa call that the Hyla scenario or the green tree frog eardrums would experience when the lungs are inflated. Here's another example of this. Here's another peak from this Fowler's toad. There's a peak here. We have something like a 4 dB reduction in the amplitude response of the eardrums in green tree frogs when the lungs are inflated. So it seems like this mechanism might work in a way to reduce the amplitude of noise from some of these other frog species that could be masking how green tree frogs are hearing their social environment. And how might this work? Well, we're thinking that this might work in a similar principle to how noise canceling headphones work. So let's say I have this little diagram here that shows us there's this unwanted noise. So this is fluctuating unwanted noise. And within the air cup of your active noise canceling headphone, you might have this microphone that's facing the outside. It's probably integrated with some signal processing circuit. And in this audio input, we have your favorite music that you want to listen to. What this processing unit is doing is it's producing the spectral analysis of the noise that's on the outside of that ear cup. And it's inverting that spectral analysis to create a waveform that cancels out that external noise so that what reaches your ear is just the music alone. So this particular mechanism in frogs, it's not a perfect thing. The frogs don't inflate their lungs and totally shut off all the other frog species that are calling. But what we think is a range of 4, 5, 6 on average dB of reduction, which is substantial for what the frogs need to hear. So how does this diagram relate to the frogs? Well, we're thinking the lungs here, when they are inflated, the lungs are acting like a microphone, right? It's recording, it's receiving that sound that's in the environment. And then that sound that's being received by this microphone or these lungs, it gets transmitted, right, through the glottal slit, through the glottis. And that acoustic energy can then reach this mouth cavity, and it's interconnected through the eustachian tube to the inner surface of this tympanum. Remember that the calls are also impinging on the external surface too. So there must be some kind of a complicated interaction here where this sort of a noise reduction mechanism might be reducing how the eardrum is vibrating in response to a defined range of sound frequencies. And that is helping the eardrums capture less of that noise that's in the environment. So just to dig a little deeper and to sort of look at what happens beyond that eardrum. This is plotted here is amplitude, sound amplitude. And here we have on the X, is sound frequency. These V-shaped curves here are known as frequency tuning curves. In yellow are frequency tuning curves from auditory fibers that have been previously recorded from the amphibian papilla, which is the auditory end organ that's tuned to the lower frequency peak of the call, which is overlapped here in black. In green, we have these fibers that are sensitive to this middle frequency range, which is also still part of that lower that lower amphibian papilla or that amphibian papilla auditory end organ. In the basal papilla, we can imagine this as a broadly tuned filter, which is situated or tuned to process the higher frequency peak of the call. So auditory neurophysiologists, we're generally very interested in what happens at sound levels that are low, right? Like what is the minimum sound intensity that would cause this auditory fiber to respond, or what is that sound intensity that would cause this auditory fiber to respond? But background noise in a chorus is actually somewhere up here, right? So at this level of noise, that is actually activating a lot of these auditory fibers from the low range, the mid range, and the high range all at the same time. And if that is the case, one solution to representing these two spectral peaks is to sort of clean up what's happening in this middle frequency range, right? So in between these two spectral peaks of the call, we're having these fibers respond to these frequencies that are in between that range. And one way to reduce the impact of this is to have the eardrum be less sensitive to those sound frequencies so that you have less of this activity that's happening in the middle between those two call peaks. And that's sort of what we're thinking of and what we're headed towards, looking at how that auditory system is actually processing the tympanal inputs to this auditory system. So I just want to summarize here. Frogs, they have lungs that are receiving auditory, that are receiving sounds in the environment. And this lung input is also internally connected with ears that are internally coupled. And we have found evidence based on our analysis that these lungs have nothing to do with improving the frog's ability to determine sound direction or the direction of conspecific calls. We have found no evidence for this, but instead we found supporting evidence for this sensory processing mechanism called spectral contrast enhancement. And this spectral contrast enhancement technique is a way to improve the signal to noise ratio of the frog's auditory system in processing the frog's conspecific calls by reducing the impacts of noise that are from some heterospecific species. So that's the story that we have so far. And I just wanted to take this opportunity to thank former lab mates and current students that have helped us work on this problem and that have also made us think about this problem in different ways as well. Thank you for your attention.
Well, thank you, Norman. Gosh, this is a really interesting talk. I'd have a few questions of my own, but I think at this point, for those in the audience, if you would like to put your questions in the chat, we'll take a few minutes and ask Dr. Lee to answer our questions.
Hey, Dan, can I ask a question while we're waiting for people to chat in?
Sure, jump in.
I just learned right now that frogs have a very similar hearing connection with their lungs as fish do with their swim bladder. I was wondering, do fish have some of these same spectral, has anyone looked to see if fish have any of these same spectral reduction abilities or capabilities as frogs do?
Yeah, and I guess the short answer is no one has looked. I think this would be the one example of looking at a non-human auditory system that shows a spectral contrast enhancement approach. So I don't know. Maybe that's possible, but no one has looked.
It's really cool. I never made the connection before listening to your talk, so thank you very much. That was very informative.
Thank you so much.
Yeah, absolutely. So I'll throw a question out there too, and Jen, maybe you can help watch the chat to see if we have some other ones come in. But so you talked about, was there 5 main species of other frogs that you looked at their calls and the potential spectral overlap with your focal species. Did those species that you focused on, were those ones that would have been overlapping in range with the population from which you drew for these experiments? In other words, I think, are these frogs you collected in Minnesota or Texas, or and then yeah, were those species ecologically relevant, I guess?
Yeah, these species, so the tree frogs that we worked on for this study were collected in Texas. And in Texas, where we collect, you often have, or there are places that often have both green tree frogs as well as the barking tree frog, which is one of the 5 that we analyzed the data more carefully with. So in our particular collecting site, there aren't any barking tree frogs, but it's conceivable that nearby there are other frog species that would co-call or co-occur within the same sort of range.
Mm-hmm. Yeah, and I guess my follow-up to that would be, would you expect, you know, across this looks like a species it's got a broad geographic north-south range. Do you think there would be some sort of local adaptation or any capacity for that in terms of, you know, a population that's maybe overlapping a group of other species that maybe had a different range of biotic interference with the calls? Do you think, I guess the question is, could there be local interference, or is a lung resonance something that's pretty fixed?
So that's an interesting question. And I think maybe one way to look at that question would be, how common is this mechanism across different frog species that vary in size and shape, right? And we're thinking that given that the couple of frog species that people have looked at, where they've described these general patterns. So the general pattern of having calls with 2 spectral peaks, a low peak and a high peak, and then having that pattern of a lung resonance that's in between both spectral peaks. And in those three or 5 species that people have looked at, they share a common ancestor sometime about 150 million years ago. So if those attributes, the patterns are derived afterwards, we would expect this mechanism of canceling noise or reducing noise to be quite common among frogs And the next thing that we really want to do is to figure out how common is this particular mechanism among frogs that vary in sizes and of other species too. But for local adaptations, we haven't looked that closely yet, so I don't have a direct answer to your question.
There's a question in the chat.
Yeah, so Michael Ainsley has weighed in and asked, what's the significance of a reference value of 5 millimeters per second per Pascal, I think? And maybe, Michael, if I didn't present that question well, you could weigh in, turn your mic off and weigh in on this one. Norman, you want to take a stab at that?
Yeah, so that had something to do with the sensitivity of the laser being 5 millimeters per second per volt. And then we had calibrated the laser measurements with a sound calibrator at 20 micropascals. And so there was some conversion that was going on, which left us with the reported units on that scale.
Got it, that makes sense. We have another question here from Andrew Simmons. She asks, how much individual variability do you see in the eardrum response in terms of both directionality and frequency response? One of your graphs shows lots of variability in the inflated state. Is this individual body size?
Let's see, individual variability you see in eardrum response. So I guess there is a considerable amount of variability, which is why we reported from something like 21 animals rather than the 2 or 3 that people typically publish with for this type of a study. And in all the plots that we do report confidence intervals that describes that variation. But the patterns of what happens to the reduction when the lungs are inflated and when the lungs are not inflated, those patterns are generally pretty consistent. You might have differences in amplitudes, but the pattern is quite consistent.
Right, thanks, Norman. Yeah, so I think that the question of individual variation is an important piece when we look at any of these sorts of dimensions of that might be under selective pressure. So,
and I guess I didn't address the body size part. Let me take a moment to address that too. So we know that when we're measuring things like lung resonance and we're keeping track of the animal's body size, the lung resonance shifts a little bit with body size. So larger animals will have a lower lung resonance than compared to smaller animals. And we also know that there are these physical constraints that also determine the frequency content of male calls. So males that are bigger tend to produce calls that have a lower frequency component to them than compared to males that are smaller. So there is this physical relationship between what male frogs are producing and then also what female lungs are resonating at for this particular species. But we really want to take a deep dive into looking at some other species where you have really big sexual size dimorphism, where you have some frogs that are huge females and small males. We really want to take a close look at how those relationships would pan out in terms of this spectral contrast enhancement mechanism.
There's another question from Kim Babbitt. Have you looked at interspecific variation between anurans that are territorial versus explosive breeders?
No, that's a great question. Yeah, and at the moment, we've really only looked at it from this green tree frog, and I think the next grant that we write will be to look at this a little more closely across species that are territorial versus the ones that are explosive breeders and et cetera. Yeah, that's a great question.
Yeah, to follow up on that, Nora, what do you think the potential is for differences between those different mating systems?
Well, I guess I would want to look at what are the selective pressures on the auditory system to develop situations where you need some noise control, right? So in these different systems, do we have situations where there's more background noise versus less background noise? And maybe if there is more background noise, there might be more selective pressure to have a more refined noise canceling mechanism, let's say, if you will. But that requires further investigating.
More work to do.
Yeah, more work to do.
We have maybe time for one more question here. We have one from Andrea again. She asked, do you think your findings can shed light on the question of why frogs have evolved two different separate auditory organs?
Good one. I'm not sure. I don't know. I'm gonna have to say I don't know on that one.
It seems like the answer is almost similar to what you just posited, where you just would have to have a lot more data on a lot additional species to see how this maps out across the tree of life for frogs. And you know, so this could be a novelty, right? It could just be these particular species, or it could be something that pops up in a variety of places, I suppose. All right. Any other questions from anybody in the audience today? All right. With that, Norman, I would say again, thank you for joining us this morning, taking your time out, and this afternoon, and joining our care community and sharing your science with us. Again, for everybody else who's still out there, first or second Tuesday of each month, we'll have a seminar, and just as kind of a primer to jump from amphibians to next month's topic, we'll be looking at birds, and our speaker from University of Texas, or University of Tennessee, Knoxville, Liz Derryberry, I think, Norman, you may know Liz, is giving a talk on how the COVID-related kind of shutdown and the change on anthropogenic noiseescapes in urban settings have seem to have influenced the call characteristics of a particular bird species. So that seems somewhat related a little bit to this talk you shared with us today, Norman, but would be our final one for the spring. So again, Norman, thanks so much for joining us, and we certainly appreciate you.
Thank you so much for the opportunity.
Yeah. Round of applause, yay!
Thank you.
Very good. Thanks, Norman.
Thank you very much.